Hello and welcome to BIO-LOGUE, a new interview series hosted by the Global Society of Genetics and Genome Biology at the University of Toronto. Today, we will be speaking with Justin Yu, a PhD student working in the lab of Dr. Ji-Hai Park at the Hospital for Sick Children in Toronto. Dr. Park's research focuses on the molecular mechanisms underlying neurodegenerative disease. And Justin here was an undergraduate student at U of T before joining the lab. Justin, welcome.
Thank you for having me.
Yeah, absolutely. We're very happy to have you. So let's start with this. Could you describe your research to us? What do you do?
Yeah, sure. So my research really focuses on amyotrophic lateral sclerosis, or ALS, which is of course, a neurodegenerative disease that causes the loss of upper and lower motor neurons in the brain and spinal cord. And this causes the progressive muscle weakness and muscle wasting. So in about, I believe, 2014, researchers, irrespective of my lab, they discovered a new gene that causes familial ALS called matron-3. And not a lot was known about this gene at the time, and specifically how it causes ALS. So my lab, of course, prior to me joining, really wanted to characterize this gene in more depth. So using the CRISPR-Cas9 system, they generated a knock-in mouse model by inducing a point mutation in the endogenous matron-3 in the mice. And they did a lot of characterization over the years of this mice, and they showed recapitulated a lot of the ALS phenotypes, such as neurodegeneration and motor defects. And actually, all these findings were published last year in October in Nature Communications, so you can check that out if you're interested.
Nice, awesome. We'll leave the link somewhere in the interview.
Okay, awesome. Yeah, so my project was really to build upon these findings, and specifically, one of the findings that they had was when they did bulk RNA-seq on the affected tissues of these mice, they found that many immune related processes were being upregulated. And specifically, if you zoom into the actual genes themselves, you can see that a lot of them were specifically expressed in microglia, which microglia are essentially just the resident macrophages of the brain and spinal cord. Mm-hmm. So my main project is really just to elucidate how microglia contribute to pathogenesis in ALS. And since I really just started my PhD, so I don't have too many findings to share, unfortunately, but I can talk about my aims.
Yeah, go for it.
Yeah, so one of the main things that I want to do is to do single cell RNA-seq of microglia. And that'll tell us many of these ALS associated microglia subtypes and their transcriptional profiles. And this will really help us to understand potential mechanisms of how these microglia could contribute to neurodegeneration. Another main part of my aim, which will probably be in parallel, which is actually in parallel of the single cell RNA-seq, is to study individual genes that are severely upregulated or downregulated in the affected tissues, and seeing really how they localize in relation to these motor neurons, as well as if knocking them down or knocking them out would alleviate or worsen symptoms, really. So those are my two main aims, I would say. Yeah.
That's awesome. That's really neat. So for people that are, I guess, wondering what a PhD student does in a day, what do you spend your time doing in the lab?
Yeah, so I spend the vast majority of my day every day just doing lab work, really. And that varies day to day depending on my hypothesis. So right now I'm doing a lot of, how can I say this? I'm doing a lot of imaging and staining of brains and lumbar spinal cords to see how specific genes localize in relation to the motor neurons, as I mentioned before. I'm also doing a lot of like RNA extraction and DNA extraction. And also, this is something specific to mouse labs, but I'm spending a lot of time at the mouse facility, as well as a large proportion of my day is just genotyping, which is a little mundane, but it's what you have to do. Yeah.
For sure. That's the strong base to get you started, eh? Cool. So what drew you to the study of neurodegenerative disease? Like why this area in particular?
Right. So To be honest, it's more of a personal reason. Before I started my undergrad, my grandpa actually got diagnosed with Alzheimer's disease, and that really showed me how devastating, in particular, neurodegenerative diseases are. Of course, all these other diseases are terrible in their own right, but specifically neurodegeneration, I found that it really strips away what makes a person. You know, in the case of Alzheimer's, it strips away your memories and your behavior. In the case of ALS, you lose your ability to move, talk, and eat, which is of course extremely devastating to both the person as well as the family members and friends involved. So that's really what initially drew me into this field. I really wanted to do whatever I could to contribute to future treatments and prevention of these diseases. And yeah.
For sure. No, that totally makes sense. That is, neurodegenerative diseases are tough to deal with.
Mm-hmm.
Yeah. So you seem to be into this, of course, for personal reasons, which is totally understandable and very respectable. What about the actual work as well fascinates you about it?
Mm-hmm. I'd say about that, the biggest thing that fascinates me is just I love coming up with hypotheses and being able to independently determine in a couple of weeks or a couple of months or even years whether I'm right or wrong. And I find that process really interesting. And honestly, I feel like I could be wrong, but I feel like that trait in particular is really important for scientists to survive in the long run. So I think that trait is really common in a lot of dedicated graduate students.
Yeah, absolutely. And what was I going to ask? Just had a question I totally forgot. They're going to have to cut this
okay.
out. I guess we'll move on to the next question. What is your favorite thing about graduate school?
Mm-hmm. So I'd say by far my favorite thing about graduate school is how much time I actually have to pursue my research. And I'm saying that because in undergrad, I've been in many labs. And the thing that really frustrated me was I would spend months and months and months on an experiment that I know that if I were to work full time, I can probably answer in a couple days or even a week tops. And yeah, I really like, of course I enjoy research. So that is probably my favorite thing about graduate school.
And how much freedom do you have over what you get to do with your day-to-day, if you will?
Like in terms of what specific experiments I can do, or
sure, your hypothesis for the day, or
right,
your focus for the day.
I think this is really lab specific, but for me, I feel like I have a lot of freedom if I have an interesting hypothesis. Of course, if it's reasonable, I can just pitch it to my PI, see if she's interested, which she usually is, and as long as it's not too expensive or time consuming, then she'll let me do it. Yeah. So my day-to-day really varies depending on what I'm pursuing at the time.
That's nice. Get to do something new every day. And how did you choose your PI, by the way?
Sure. So I was really interested in joining molecular genetics in particular because honestly, because I did my undergrad in it. So I knew a lot of the faculty members, and I think genetics is really cool. And of course, as we mentioned before, I really wanted to study neurodegeneration. And to be honest, molecular genetics might not be the best place to study neurodegeneration because there's very few PIs. Probably physiology has a lot more, but so I didn't have that many PIs to work with, but I knew J.A. Park because I went to Walk to End ALS, just a fundraiser, and that's where I met her for the first time. And I went there specifically because I worked in the Pearson lab at the time. So I knew a lot of the people in her lab, and I've heard a lot of good things about her. And I thought her research in particular was really fascinating, specifically how she works with mice. So I'd say that's honestly the main reason why I joined her lab.
Right, totally. And you mentioned that you worked in quite a few labs in your undergrad, which is cool, very cool. And U of T has a bit of a reputation of being a tough, soul-breaking school. So I guess my question is, how did being an undergraduate at U of T prepare you for the demands of graduate school?
Right, yeah. I would have to agree. It's very soul crushing. So I think because of that, it prepared me a lot because it taught me a lot of good study habits and specifically how to study efficiently because you don't really have that much time because you have to juggle so many courses and extracurriculars as well as research.
Mhm.
So yeah, in terms of that, I think it prepared me really well. And specifically since I've been in a couple labs,
Mhm.
the skills that I learned in the labs definitely translated to what I'm doing today.
Right. Totally. What extracurriculars did you participate in?
So I'm probably not the best person to ask this because my extracurriculars are really lacking in my resume. There's only two real notable things. I volunteered at the Toronto General Hospital for just one summer in my second year.
Mhm.
Or no, in my first year. Um, I was just I was a navigator. I was just helping people find specific places of the hospital.
Mhm.
Um, and of course, last year I joined GSGGB, which was a fun experience. But I personally I think if you want to pursue graduate school, research research experience is by far the most important factor. Of course, you should do extracurriculars. It'll help your resume look better. But if you want to do grad school, you should try to join a lab as soon as possible.
Good. And how did you get your lab positions? Through what avenues did you find your way in?
Sorry, you're lagging a bit. Could you repeat
Oh,
that?
sorry. Yeah, sure. So how did you get your lab positions in undergraduate school? Like what avenues did you get in on?
So in my second year, uh, I'm sure you heard of the Research Opportunity Program, ROP.
Yep.
That's yeah, I applied to as many labs as I could using that um, using that program, and that's how I joined my first lab in second year. And so I joined Dr. Carol Sherman's lab in Sunnybrook,
Mhm.
and I think once you join your first lab, it definitely helps a lot to your next lab because having any research experience looks really nice, especially as an undergraduate student. So in my third year, I got I got a position in Dr. Pearson's lab, actually through a work study because I wanted to dedicate less time and more time to study. So I just
Mhm.
did a laboratory assistant through a work study position in Dr. Pearson's lab, and then I ended up contacting him to do a full-time summership in his lab. And then in my fourth, and by that time, I feel like I had a good amount of research experience. So in my fourth year, I just reached out to Gabrielle Boulian,
Mhm.
and because her research focuses on Alzheimer's disease, which is just a big interest of mine. And that was not in a specific RP program or work study. I just contacted her and actually, no, sorry, I lied. That was I contacted her initially, but uh, it ended up being a fourth year MGY project for me. Yeah.
Nice. Um, and I guess on the topic of undergraduate research, what advice would you give probably a first year student, maybe a second year student who's looking to get into a lab? Like how could they go about it? What should they do?
Yeah, that's kind of hard. I think because it's really hard to know what research fascinates you, and even whether research at all fascinates you if you've never done it before.
Totally.
Um, I'd really say if you're at U of T, you have so many opportunities. I'd say just to give it a try in terms of research,
Mhm.
in terms of what interests you. I don't know. That's a bit difficult. I think
Just
whatever
sort
you
of,
found interesting in first year courses or in high school, I think,
yeah, just try it out.
yeah, just try it out. You can find research in revolving those topics.
Totally. And like you mentioned, research is not for everyone.
Yeah,
It's its own interesting situation.
definitely.
Yeah, for sure. And actually, what is the hardest thing about being in graduate school for you?
Right. So I mentioned my favorite thing was how much time I could spend on my research. So ironically, the hardest thing is that I have to spend so much time on my research. I feel like,
Classic.
yeah, graduate school in relation to undergraduate, it's a lot more of a time commitment. I feel like this is definitely department specific and lab specific, but I feel like if I'm not working like 10 to 12 hours a day, I feel like I'm just falling behind. So
Yeah.
that's by far the hardest thing. Yeah.
Uh-huh. And do you find it sort of hard to move on from undergraduate student to this sort of really intensive graduate student, or well, graduate program environment? Was that a tough move for you?
I think it was tough. I think the research itself I found really interesting, so I really enjoyed doing that. But of course, you still have to take coursework in graduate school. It's nowhere as much time as undergrad, but you still have to take these courses. And I feel like I had a hard time balancing research and courses, at least in my first semester.
Mm-hmm.
Probably like online Zoom courses and COVID contributed to this, but
Just a little bit.
yeah, just a little bit. But yeah, it was a bit of a struggle in first semester, but now courses are starting to ease off, and I'm enjoying it a lot more. Yeah.
That's great. What are your plans for the future after graduate school? Which is, I know it's the $1,000,000 question that nobody wants to answer, but what are you thinking?
Yeah, so I definitely want to stay in the field of neurodegeneration. How specifically I can stay in this field could vary, but I'd say probably my top choice right now is to do a postdoc after I graduate. Probably in another lab. I'm thinking the States. If I have exciting findings, hopefully, then I could join a lab in the States, hopefully Ivy League school. And
nice.
yeah, but to be honest, actually, yeah, to talk more about that, I think it would be cool to start my own lab. Of course, this really depends on how successful my career is at that point, but I think it would be cool. But to be honest, yeah, I don't think too much about the future. I try to take it like one step at a time. And I definitely have a lot of time to think about this, since my PhD will probably take like 6 to 7 years.
Wow, 6 to 7.
Yeah.
Nice. That was actually about to be my next question is, what are your thoughts on the whole, there are no careers in academia sort of debacle?
Yeah, I think life science is really unfortunate in the fact that it seems that you can't do much with an undergraduate in life science. And
not to discourage anyone.
Yeah, not to discourage anyone, but sorry.
Yeah, no worries.
Yeah, academia is really tough, especially with the funding situation and everything. But sorry, what was your question again?
Oh, don't worry. I was just asking, I guess essentially the crux of it is, what are your thoughts on what people are saying that, I mean, they've been saying this for quite a few years, but the idea that there aren't very many positions in academia.
Right, yeah. Yeah, it's definitely competitive and challenging. So I would only recommend academia if you are genuinely fascinated about research, because otherwise they will crush you.
Yeah,
but
don't do it for the doctor title, people.
Exactly, yeah, just go to med school.
Yeah.
But yeah, but if you really are fascinated in research, it's a really good career to go into, honestly.
For sure. You get to do what you love every day, more or less.
Yes, yeah.
Oh, that's great. Well, Justin, thank you so much for joining us. I guess that's going to be the end of our interview, but we really appreciate your time here, and I hope some of the viewers are getting some good insight into what it's like to be a PhD student. So thanks again.
Yeah, thank you very much.
Bye-bye.
Bye.